Welcome to our presentation on protein synthesis. The first step of protein synthesis is transcription, the making of the message that's going to be sent out to the factory to build this specific protein. Now, let's just stop for a second and think. If we are trying to make a report on penguins, you wouldn't go to the library and take all the books out. You wouldn't go to the library and take even one whole encyclopedia out. You'd probably go to the P section, the P book, take it out, find the page on penguins, go up to the desk and ask them to make the copies. That copy is like the message that's going to go home with you or going to go out of the DNA, out of the nucleus, to the factory, the ribosome. You don't need the millions and millions of other plans for other proteins to go out there with that. You just need the specific plan for that one protein that needs to be created. So we need to make a copy, or in this case they call it messenger RNA message, that from the millions of genes on the DNA, we need to make a copy of one gene and send it out to the factory. Why must living things make new proteins? There's many reasons. Uh, all the traits, the eye color, hair color, uh, that make you you are built up according to the plans in the DNA. And there are millions and millions of enzymes that control all the chemical reactions of life that are created, the information stored in the DNA and then created at the ribosomes, the protein factories, by this message, through this message. These enzymes are the proteins. That's what I just mentioned that control these things. And they are proteins and they need to be made. As their supply diminishes, more of them need to be made. And repair structures like tissues, organs, we're built on essentially protein and bone. So proteins are very important. What are the raw materials for proteins? Unlike starches and cellulose, that's just sugar, 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 chains of sugars. It doesn't really matter. They're all the same. But proteins, the raw materials of proteins are very different. This is what makes protein synthesis so complex. The order and amount of these materials is very important. If you make a mistake by one in 500 or 1,000 of these little steps, it's not that protein anymore. It's garbage. So all creatures must eat protein like beef, chicken, fish to make these proteins, to break down the proteins into these basic amino acids. Yes, they're consumed, broken down, and then absorbed as amino acids. And these are the building blocks of the proteins. And there are a number of amino acids, and they're all different. And that's the key to protein. The order and sequence of these amino acids is vitally important for that protein to be the protein that you needed, that you need to create. And that's where the DNA comes in. It contains the instructions or for the order of these proteins to be made. Amino acids are then assembled in the exact right order, in the exact right length to make that specific protein for that organism. One little difference and your hair isn't curly anymore, it's straight. Or your hair isn't straight anymore, it's curly. One little amino acid sequence different makes a difference between people with curly hair and more straight hair. Genes are the section of the DNA that codes for one protein. That's really important. There are millions of genes on the DNA, but we need to make one protein. We need to activate or work with this one gene site, a tiny little place on the DNA. DNA is tens of millions long. A gene might be 5,000, 10,000 nucleotides long. And then in the whole DNA thing, um, only some of the nucleotides actually contain heredity information dealing with you. They're called exons. Those that don't contain heredity information called interons, the interspace. We're not sure if it's just holding places or it's old DNA that we no longer use as we have changed from one species to another. A couple of differences between RNA and DNA. RNA is ribonucleic acid. The real only main difference is DNA is deoxy. Is missing an oxygen. It's deoxy, like decapitation without a head. Well, with deoxy without an oxygen. So ribonucleic acid, the sugar is the everything's almost exactly the same in that nucleotide, except the sugar. Remember phosphate sugar base? The sugar in the middle has an extra oxygen on it, so it's RNA. There's three different main differences between DNA and RNA. RNA is a single helix. It's not a double. It doesn't have a secondary side. It only has the side with the codes for the information. And as I just said, RNA contains ribose sugar instead of deoxyribose. And here's a key thing. There's a different base. Uracil replaces thymine. In RNA, there is no thymine. So we're always doing, we just learned that, A goes to T and T goes to A. Well, now A will go to U and U will go to A in RNA. So if it's DNA, it still has thymine. Once we start making these things in the RNA part of the cycle, there's no thymine. It's always replaced with U. Types of RNA. There's three types of RNA. Two of them is what we will concentrate on in this course. Messenger RNA, that piece that we're going to make in the nucleus, is part of transcription that will move out to the ribosomes, the protein factories, and create the protein. Transfer RNA, which is, you'll see in the next unit, helps to make the proteins. And ribosomal RNA, which is sitting out there on these protein factories, waiting to be used. Amino acids are the building blocks of proteins. Humans use 20 different kinds of amino acids. Some of these we can create, but there are what we call 8 essential amino acids that we have to consume in a healthy diet in order for them to be there for the process of protein synthesis. So, steps of protein synthesis. We're not going to see all of these in this unit, but you start with the DNA. That's where it begins by the gene that's needed gets activated. And then transcription is the copying, the writing of the DNA code to make a RNA molecule, single-stranded RNA molecule. That messenger RNA then moves to the ribosome. That would be the end of this unit, but just as to keep going with the overview, then we're going to read that DNA code, which is now a messenger RNA code, by the truckers RNA in molecules, bringing in the amino acids, and the transfer RNA will bring in these amino acids then in just the right order to make this protein as commanded by the DNA. The whole process of protein synthesis is to make a protein. First step in transcription, the helicase opens up the DNA, but only at the gene site, not the whole way like in our DNA replication. It's only at the gene site. And then the RNA polymerase opens up the molecule at the promoter on the side that the gene is on. Remember, DNA has a gene side and a complementary side. Complementary side is only there for replication. We are not replicating. We're going to make a messenger RNA. And then this RNA polymerase brings in these RNA nucleotides and plugs them in, C to G, G to C, A to U, and T to A. Remember, there's still T on the DNA side of these pictures. And we're starting to make a strand of messenger RNA, which will pretty much be exactly like the complementary side of the DNA molecule, except you can kind of see there at the top of the green one, There's a C, but then there's a U where there would be a T. So you can kind of see as you go along, C, U, C, U, U, that it's G, T, G, T, T. It's almost the same except the U replaces T. And we're making a strand of a message. That's transcription. Then, like I said, the RNA polymerase brings in the free RNA nucleotides and they get paired up. And in this case, it's green color. The browner color is the DNA. And we're getting a message made. And remember, U replaces T. The end result of this long, maybe 7,000, 10,000 long strand of messenger RNA will be the kind of like the negative code for the DNA coding for the protein that we'll learn about in translation. Now, the fully completed messenger RNA leaves the nucleus, goes to the ribosome where translation will take place. So, transcription is now done. This is the process by which a messenger RNA molecule is created in the nucleus. Transcription occurs always in the nucleus and ends when the messenger RNA leaves the nucleus by RNA nucleotides being brought in to match up to the gene side of the DNA molecule only at that gene site. The DNA only unzips at the gene site, allowing RNA polymerase to bring in RNA nucleotides, matching them up perfectly to the DNA code. The only way it could be, G can never hook up to A. So we're making a strand. It ends up being kind of like a negative of the DNA, and it goes out to the ribosome.